Good morning, everyone. Thank you. The first thing I would like to ask, because I did this 4 conferences ago, how many of you use Bioinputs here, please? Raise your hands. Well, more than 4 conferences ago, and that is important. It is more than interesting being in this world for a number of years. This is a mind-blowing subject. It's hard to explain. There are more and more people joining this move. But I saw one of the presentations on the first day from the Brazilian people, and they talked about the production gap and what are we failing to see. And I think that biologics, I understand from what I've seen in the last few years, bioinputs in general are sort of moving the foundations of the things that we are messing a bit. Everything is Q now, so we don't start increasing our production and raising the production floors, will be broken. I would like to thank the panelists, and I think it is very good to talk about this. Those who are not using bioinputs, I hope I can convince you the reasons why to use them, because what we can see is that what is helping us increase the yield. They come along traditional fertilization, and it's not substituted by, it's not that we're trying to substitute for it, but we're trying to see how we can integrate them, how we can use them to increase the yield and to do away with our production gaps. I'll leave you with Walter, who is a co-founder of Bactools Biotech, a graduate in agricultural engineering. To start understanding how to boost production by adding bioinputs, we need to understand the microbiome of the plant. The microbiome is a multicellular ecosystem that goes along the whole plant with a number of microorganisms in a symbiotic or non-symbiotic bond with the plant. This is something we know. Nodule in soybean, and if we pay attention, we will see there are other microorganisms associated to the plant. Something we need to see is that they are not randomly on the plant, even when there can be opportunistic microorganisms. For a while, gene clusters have been found in the plant to synthesize chemotactic signals that attract a number of microorganisms that are beneficial to the plant, as well as sequencing and genomic sequencing tools that we are using now, where we can use not only soil samples, but the whole plant. And we find there is a central microbiota associated to every type of plant or crop. A wheat plant in Argentina has many well-preserved microorganisms vis-a-vis other plants somewhere else in the world. So there is a close bond between microorganisms and plants, and that is very important. On the other hand, in that community, there are microorganisms that are beneficial, that are captured or called somehow by the plants, but there are also neutral microorganisms and others that are pathogenic. So when you look at that group of plant-associated microorganisms, you can say, what is it going on? I may know, for instance, characteristics of certain microorganisms, a bacillus, anti-fungi, and control a fusarium. But when those microorganisms meet at the plant, who wins? The fusarium, the bacillus, and that is an emerging property of that ecosystem that we scientists find it hard to predict. It is an emerging property in that group of microorganisms, those microorganisms in the rhizosphere, and one will prevail. The Fusarium may win and sicken the plant, and the bacteria may win and colonize the root and fight that pathogen. We cannot predict, but we try and tilt the balance by using bioinputs, trying to favor that certain microorganisms be present from the root, from the birth of the plant, which are beneficial for the comprehensive development of the plant. What I'm going to show you now are two developments of the company that are working very well. One is a consortium of 4 bacilli with different characteristics, and another product has a very good capacity to mobilize and use sulfates from the ground. The consortium of 4 has growth promoting activities and biocontrolling activities. This is the average of the results we are obtaining in different crops. Seasons 23, 24, and 24, 25 in different geographies. Gray is the Czech specimen, then orange is the treated specimen. We get 7, 8, 9% yield in seed treatment. What we are doing as well is the foliar use of microorganisms, which has a big impact. I was at Utria's presentation. That is a very good microorganism for the development of plants, and with a noticeable increase in yield. On the other hand, the other bio-input that we are applying with very good results is bacteria that solubilize phosphorus. This is a Bacillus amyloliquefaciens. Gray is the Czech specimen, orange is the treatment. Now, as we are measuring solubilization capacity or phosphorus mobilization, what you compare in yellow and in blue, 100% of the recommended dose for that plot with SPS, and in yellow you can see 20% less of SPS dose, but with the seeds were inoculated by the phosphorus mobilizing bacteria. You can move 20% more bioavailable phosphorus to the plant and cut down a bit the chemical pesticide use. You can see 10, 12% increased yield, which is the global average of bioinputs. Let me tell you what we're doing at Bactools. We have moved more going to our bacteria genetics to understand more what is going on. The interesting thing about this is to look at it from the number of genes you are contributing with, growth promoting genes when you treat a seed with a bioinput. For instance, we sequence the bacteria of the consortium of 4, the one we showed you. There are, before, and each strain has 4,500 genes, and between 1,000 and 1,500 are called growth promoting terminals. They are associated to a growth promoting end. In this case, 4 strains contribute with some 6,000 growth promoting genes to the seed, and each strain part of the consortium have single genes, and they have some characteristic of growth promotion. The look that we have from Bactools is to activate bacteria without altering the genetic makeup, but to genetically regulate a number of things. For our bacteria to have those genes, maybe it's more for us, this information, but to have these associated genes does not mean that they are expressing. This is the first step. Gene sequencing to see what tools we have, and then we go to other proteomics, transcriptomics to see what is being done. But our objective as product developers is to have the bacteria that when they are applied, they have an active genome. That is very important for us, and that can be the departing point to jump that 10% increased yield that we find in biologicals today, and to take us up a new step. In general terms, to wrap up this part, bioinputs add living functions, and that is something that we should highlight. Promote soil health, stimulate plant defenses, and that is the activity of bioinputs. Biotechnology does not replace anything but supplements regular or typical activities, improving the assimilation of phosphorus in the plant, being a supplementary tool. It's important to have a good diagnosis of your plot because not all bioinputs have the same applications. Some move minerals, some move other things from the soil, some improve plant defenses, some are biocontrolling inputs. They are specific to the activity you want to promote in each crop. They are not a future promise, they are a reality today, and I've seen this at other presentations here as well. Yield is increased, and it's not choosing between chemicals and biologics, but using the best in each one of them and to have them supplement each other. Thank you very much.
La próxima, por favor. Perfecto. Vamos a seguir con...
All right, we will now listen to Gustavo Gonzaléz Anta. If you don't know him, he is the CEO of Indrasa SA. He has a PhD in nitrogen fixation, biological fixation, and he has a PhD in soil biochem. Thank you for staying until the very end this morning. What is this mess you see on screen? Well, I'm trying to provide some context for us to realize what to do to enhance yield in our production system and what bugs may help us. And there's very many bugs out there. Some are very specific, but there are very many of them that may allow us to increase productivity in our production system. Let's start at the very beginning. Walter was talking about seed treatments, and we know that that's where we define the result of our cultures, of our crops. So what do we need from a seed? We need nutrition. So this is soybean, which is what we have the most in Argentina, but it can also be applied to other crops. For soybean, for soya, I need to work on the symbiotic relationship with different bugs like Rhizobium hyponicus that allow us to fix and has other PGPR capabilities so as to have the right growth, forgetting about nitrogen sources. This is not traditional, but it's been measured, and it ensures about 8 to 10% more yield. If we don't do this, we will lose that improvement. We were saying Q is more important than T here, but also having last generation sources is good. We want a healthy emergence, and we will treat with fungicides. Here are a few kids, trichoderma and bacilli, that allow us to ensure that those seed pathogens or soil pathogens will be controlled. Now, one more thing that I'll talk about later. we usually use biofungicide bugs with disease control. In fact, microorganisms produce molecules to control pathogens. If I talked about thiazoles, benzimidazoles, purines, and carboxamides, all of them, give or take, all of us will understand what we're talking about. But there's purines, surfactants, etc., molecules that control different pathogens. And what's the difference? Basically, there's bugs producing them, and as a result, it's the molecules producing the control. So what we want to achieve when we treat a seed is to have a good establishment of the crop, a vigorous one. So there's also a number of bugs that will allow us to improve root development, to explore different areas of the soil. So the different crop cycles help us from scratch to become efficient with nutrition, pathogen control, and growth control. And our crop keeps growing. There's a vegetative and reproductive cycle for our crop. We have seen major progress in connection with formulations. We can now have bugs that will live much longer than in the last century. Therefore, again, what will be the problems during my crop development? There's two: abiotic and biotic stresses. Biotic stresses, if we use different microorganisms, biofungicides, for example, will allow me to control or to associate certain molecules. And instead of mixing with liximerazole, triazole, and etopiramide, I will mix a triazole with a microorganism, normally a gram-positive, but others as well, Pseudomonas, or gram-negatives, that will allow me to check the disease and achieving two things: controlling the disease and decreasing the likelihood of resistance, which is a problem in weeds, for example, and which we are thinking of now when it comes to fungicides. And then insects in subtropical, tropical areas in particular. So we have introgressed BT, which came from a bug. We put those genes in a plant to make our control more efficient. But also, thinking broadly, this would be a bio input coming from an organism that was controlled and which was made to express the resistance. In the case of BT, we have seen major increase of alternatives to control new pests. And in tropical areas, there's other bugs which we know because we had to fight them, like Dalbolus mites and others, that have been our foes, and also Lobelia, Metarrhizium, Fusarium, pathogens that may be connected to bugs in order to produce control. So, all along the development of our crop, from settlement to reproduction, controlling biotic stresses, diseases, pests, the image needs to add the other stress, the abiotic stress, the more difficult one, maybe. And if we want our crop to develop, we could bring other organisms. But let us not forget that there's a before and after seeding. I don't have much time left, so I won't talk about bioherbicides, because it's a whole new chapter. But we know about carryover problems, a problem that restricts our decision-making. Thank God we have bugs that can use that complex carbon source to break them down. But here are Pseudomonas of different kinds and characteristics that will help us decrease the use of phytosanitaries from our soil to enhance our production. And then there's also a different story. What happens after harvest? Because maybe we did everything fine, but then we stock it poorly. So there's toxins that when I put my product, my effort product in, and I silage it, then I have the endobacilli to solve this problem. And for the Q&A session, how can we improve our productivity? And when we think about it, we think of one of these things, but a bug may participate in all of them and not just substitute some phytosanitary products, but also, and the word that is the name of the game is efficiency of use to try to reduce resistances. And as a result, integrating all of these concepts would be useful. Thank you very much. You can ask your questions later.
Bueno, muchas gracias, Gustavo.
Thank you very much, Gustavo. The presentation is really nice, quite open. I hope questions start coming up. And now we'll give the floor to Rodrigo Martin, an agricultural engineer involved in plant Agrotecno efforts in over 65 countries, and they are in the 5% of the best companies worldwide as to sustainability. Rodrigo, you have the floor. Thank you very much. Well, I am the father of 7 children. You missed that. The most important thing. My latest child was born yesterday. Rodri, we don't have your slides, I'm sorry to say. No problems, I had sent it. Anyway, it's my pleasure to be here. I would like to thank all those that are listening to me and those that are on the other side of the connection. Aapresid is a spectacular association. Martin and I have been working in the network of biologics, talking about sustainability, regeneration of soils, heavier, lighter soils, vertisoils in the north, sandier, clayer, and so we want to make the best of the characteristics of the environments, and that is what I wanted to talk to you about. I wanted to talk about the diagnosis. Cole is here, Walter, Gustavo, Maxi have talked about the options. They have opened the toolbox. What tools are we going to use if we don't have a prior diagnosis? Actually, technology has moved forward so much for diagnosis. We need to know what is going on each plot. Technology lets us use in 3 times 3 pixels. It's like going every 9 square meters on the plot and review and conduct an analysis on that piece of land. I'm going to talk about diagnosis. We're highlighting this a lot because the tools in Argentina are spectacular. There are companies with very good developments. Agrotecno is one, but there are others. We need to choose the right tool. We do know that crops live of the reserves in the seed in on the first 20 days. And depending on accumulation, that will have an impact on the growth the next year. Are we working on plots that will have seeds for the next year? What's the rooting capacity, the reserves, pH, how hard the soil it is, microbiological activity? All these questions may be sorted out very quickly today. Let me tell you our experience in Chile with intensive agriculture. They have lesser surface areas available. You need to look at many aspects. So they work on the first portion of the crop, that is to say, the root. I'm sorry the presentation is not here. I was going to show you a couple of pictures of diagnosis and different soil views, where all agricultural engineers, and first of all, we want to look at our soil, our roots, at the quality of the roots, their health. It can be wheat or rapeseed or olive or pistachio, all. But that is all about this. And now we can see whether the root is active or not, the microorganisms involved. From Agrotecno, we are working to feed microorganisms with our products. Fulvics, short change, organic base, because if we feed them the microorganisms, we will sort the problem out. I can talk to you about tea. The Argentinian tea has a pH 4 soils, very low pH, constant defoliation, and they may be not looking about root regeneration. There can be problems with nematodes. It happens in Argentina. This can be the case with tomato. How can we live with those microorganisms to get a nice root so as to be as efficient as possible? I'm talking about the plant and absorbing nutrients. And they are work with the correction of their necessary nutrients to tap the resources and on-site and for yield to hike. I can assure that that is the case. You will see that there are many and many more options of products, but if you know what you want, I want a good root system with these characteristics, I'm sure 3 or 4 options will appear and you will get your root. In soybean, you know that it is indeterminate after the emergence, and a healthier plant will behave better in the light of pathogens and fungal complexes. Then you need to have a good blooming of the plant to contribute to the yield, a good formation of the fruit. This is not an isolated technology item. It is a productive system, a production system, starting with the seed and ending by remobilizing all the resources to the seed and increasing weight. We need to take into account a farmer profitability. So much more today, not only in intensive but also in extensive crops. So working with the efficiency of nitrogen, potassium, phosphorus, manganese, molybdenum units that the plant can use is critical. It's not quantity but quality, the quality that can be made use of by the plant. And when I put the variables in place, the fructification, the blooming, everything gets online. We work a lot with pistachio, which is hip now. A lot is being said about pistachio, but if you see the place where it's planted, the conditions are quite adverse. They grow in the desert, stony soils. You need to see this high value added culture needs, crop needs. But it also happens in rice in the province of Corianes, where we see there are productions over 14,000 kilograms of rice with good nutrition, where the word out there in the past was 9,000, same thing with peanuts. We are breaking yield roofs, or ceilings, better said. But in order to put the resource in place And to make the best of your consultant, you have to get a good diagnosis. And a farmer or an engineer that started with their diagnosis, they are already tackling their problem, and serious companies will be available for you to give you the right tools. Thank you very much. Impressive. A double round of applause because he didn't have his slide deck.
Bueno, están llegando preguntas. Creo que Rodri que acaba de decir algo que me parece.
I think, Rodrigo, you said something several times, and I think it's important to say it once again. When I started using biologicals, it was because I saw it used in Chile in intensive crops. The difference between Chile and Argentina in intensive crops is enormous. The average for nut trees is 4,000 kilos here and 6,000 to 8,000 in Chile, all thanks to biostimulation and working on roots. Rodrigo, your experience, I mean, is great. Everybody uses a magnifying glass in this sense, and we don't. When you do the soil pits, the test pits, you really magnify your look. And this is something that we tend to forget because we look forward, push forward, but I think this brings value to our possession. We were listening about bioinsecticides. We need to think of damage control, understand bioinsecticides. You know, the use of Bacillus thuringiensis, they said 1.5 liters for R1. That was too late. The worms would eat your plants before that. So if we started thinking, let's split the dose in 3. Use your common sense, that is, and try to question what we do. We need to assess our chemicals as well and our biologicals. This is the nature, and this is what our forefathers used to do.
A question to Gustavo. What to control abiotic stress? What microorganisms do you use? Or you're going straight to the bone? We've been working quite focused on a series of bacilli. We've worked with Psychrobacillus, Revibacillus, Licenibacillus, bacillus themselves, the line that Walter was talking about, with microbial consortia of gram-positive bacteria. We have a better use of the water, but also, and something we are interested in, is better radicular development in depth. I have an emblematic case. You may know the Paraguayan Chaco region. That is 600 millimeters rainfall, hot temperature, a lot of evaporation and transpiration. And when, in the light of those environments, you use this consortia, is, as we said, night and day. I would like to wrap up my answer by saying that rather than the bug, the name, you have to look for the function, because even when we work and have obtained good results with this group of gram-positives, there is a multiplicity of other microorganisms that if we manage them carefully and ensure their survival at the time of application, we can get the same outcomes. There are other bugs that can help us there as well, but in our case, we work with that. Thank you. This is a question to Rodrigo. Organic matter. That is a big bag where we put anything. And I think it is important to talk about differences, time of application, Is it the same to use a composted or non-composting chicken manure? Stabilized, non-stabilized, algae, amino acids, when, how? Would you please expand on that? That's a great question. I'm going to open a parenthesis there. In Argentina, we don't yet know, or I think there is a biostimulant act coming out to properly record everything. They are fertilizers or amendments or organic matter, too comprehensive a concept. In that bag there are many things, fallback, short chain, etc. But not to make you dizzy, anything that will let the plant be more efficient in absorbing such or such nutrient. At the end of the day, we can work with very sandy soils, pure sand, where we will add these organic matters so that they bring to availability all the macronutrients, and amino acids will help the plant to pick them up. Let's go to concrete cases. It happened with a very high volume production grapes in the province of Mendoza. When we had the test pit, we didn't see roots. And so everybody said NPK is missing. So they produced 40,000 kilograms of grape without NPK. The thing is that it was there, but there were no roots. And that is corporate know-how. The quality of the formulation and its stability are key factors, because we're working with amino acids and many compounds that are volatile, and so we need to keep them a long time, generate the radicular system, and from there start nutrients. We talk about zero NPK, that's too much. But it was not needed because it was there, and the diagnosis did show it, but they needed uptaking. That's the case, Maxi. You need to differentiate. Remember, manure and all. That's very interesting, but sometimes they bring resistant weeds or nematodes, and you have to keep an open eye. So apart from logistics, I believe that with good amendments, with new treatments with organic matter, you can get good results. Thank you, Walter. These bioinputs you showed, do they stay in the soil, or it's just the plant's life cycle? Do you need to add them every year? Oh, what a question. These bioinputs typically create a spore, and maybe it has a permanence in the soil. You reminded me of the story of what was called dead plots that had anthracis, bacillus, spora, and nothing grew there because the mechanism stayed there. So the working with bacilli has that advantage. They get into the soil little by little, and they regenerate the microorganisms that have been moving away. The advantage is that they have many metabolic routes that are very important and can be used by plants. Starting the use of bacteria to generate the soils with bacilli, with metabolic reserve, is very important to recompose the health of the soil and little by little re-establish those microorganisms. Thank you very much. This reminds me that yesterday was the Veterinarian and Agricultural Engineers Day, so congratulations. And I think that it depends on the area of the organic matter content, the area. There are areas where people say they don't need inoculant for soybean, and they don't use it. And if there is a lot of response to inoculants, you know, the word we always say, we engineers say, is it depends. Well, no more questions. Thank you all. If you want to add anything else. I think that the back and forth you shared is very important. From this side of the street, having a back and forth with the farmer, because you think of something, you go to the plot and see something else, but the ones that know their plots are the farmers. It always helps improve performance. What Gustavo was saying before, microorganisms have many different characteristics, and so what Rodrigo was saying, diagnosis. What do you want to do? What are you looking for in your plot? And there are microorganisms with specific functions that can help you better than others. I hope I have been of use. And last comment as to universities, Valencia, Granada in Spain, our own R&D teams are already working with genes. As Gustavo and Walter were saying, there is an increase in the number of microorganisms and beneficial fungi when there is food, when there is a benevolent ecosystem and they express all the necessary benefits required by the crop. Thank you. Just let me wrap up by saying something about what Walter said. There are 10 billion bugs per gram of soil. Imagine how they can be vehicles by applying something at the plant or in the furrow even. In this symbiosis with crops, you know the sources of carbon in the roots. When the crop has been harvested, that source of carbon goes away, and there are 10 billion bugs that have been there for million years and will not leave their ecological niche to be attacked by what we bring. And there is a whole year in the middle where things happen, as our president says. Temperature changes, moisture changes, crops change. So at the time to use this type of technology, we need to have enough to get the agricultural effect that we are after. Even when there are some bugs left of those that we have added to the soil, it's also true that they are not in the number or in the position that at the time of planting are required to express the crop's potential. To wrap up, I think that on one of Walter's slides it read the soil as a holobiont. And we need to understand that when we are planting something, we are causing disruption. And we need to tend to balance for the system to accompany us and to reach that balance. That requires reestablishing the soil, biotic, abiotic components. We've been focusing on biotic components, but in year 2000, the publication in the States came about the abiotic stress of crops, and I believe that that can be the place where we can make a difference. And in years like this one, where we are all trying to adjust our behavior because we know what is going on with our figures, and we know we want taxes to be reduced. And so we say, let's be better at farming. And I think that is the path. We need to get better at that. And from Aapresid, and that's why we have this panel here, we believe that bridging the gap can be in the hands of inputs. Thank you so much for attending this conference.